Hi, my name is Tsuda. I am a fourth-year bioengineering and pre-med major currently interning at the Focused Ultrasound Lab at Brigham and Women's Hospital in Boston through Northeastern University's co-op CaNCURE program. If you or anyone you know has ever been injured, you may know that there could be lasting effects on your body known as chronic pain. Even now, I currently have a broken hand. Damage to the nerves after an injury can leave them inflamed and dysfunctional, which could cause something called neuropathic pain. Neuropathic, or chronic pain, is a global health crisis that affects 1 in 5 adults in the US alone and is the #1 cause of disability and burden across the globe. Now, why am I talking about pain when my project is on focused ultrasound and cancer research? In order to answer this question, let's trace things backwards. And when I mean backwards, let's explore the place where we actually feel the pain, the brain. As we know, the brain is a complex machine intertwined with neurons, glia, and blood vessels. Today, I want to focus specifically on the interface between the blood and the inside of the brain, cleverly named the blood-brain barrier. The blood-brain barrier is one of the most selectively permeable membranes in the whole body. Endothelial cells connected by special adhesive proteins create tight junctions and make it incredibly difficult for proteins to pass through from the blood into the brain. Moreover, drugs that do currently perfuse into the brain from the vasculature are non-specific to the brain regions, meaning that it just goes everywhere. Now, imagine if I'm experiencing chronic pain in my right hand and there was a way to increase the pain-relieving effect of a drug into my hand. What if I told you there was a way to increase localized disruption of the blood-brain barrier to target a specific region in the brain and potentially raise drug efficacy into the brain? Enter focused ultrasound. For our focused ultrasound experiments, we use rats that are in a chronic pain state and cause disruption and their BBB, followed by systemic injection of a pain-relieving drug. The two results that we analyze from the rats are functional MRI data and behavioral analysis. The rats are put in a chronic pain state after undergoing a spared nerve injury surgery, which severs a major nerve in their leg and causes them dysfunction in their leg over the next several weeks. For the focused ultrasound treatments, the rat is anesthetized and the head is placed on an ultrasound transducer. Though it may sound like the ultrasound itself is disrupting the BBB directly, in fact, the expansion of special microbubbles excited by ultrasound energy is what causes localized BBB disruption. These microbubbles are injected into the rat and clear out of the rat's system within minutes. The disruption leaves the BBB open for several hours and is still open when we give a therapeutic drug into the rat to relieve the neuropathic pain symptoms. Our specific target in the brain is the somatosensory cortex, where the brain receives and processes sensory information from the body. We hypothesize that by disrupting the BBB here may increase efficacy into this region, further assuaging neuropathic pain. I mentioned before that we analyze the effects of this treatment through two methods, functional MRI and behavioral analysis. Magnetic resonance is an important tool that we use to visualize how the focused ultrasound is affecting the brain. And the two types of MRI we take are structural and functional. First, we take high-resolution structural MRIs after treatment to confirm that there is no structural damage to the brain. On the other hand, functional MRIs are a series of lower-resolution images that visualize the blood flow inside the brain over a period of time. After processing these fMRIs, we are able to see how blood flow to certain regions in the brain positively and negatively correlates to other regions. With this data, we hypothesize that drug therapy with BBB disruption may be changing pathways in the brain, which is information that we can use to fight neuropathic pain. We also conduct behavioral testing in the rats by applying mechanical stimulus to the injured limb. Normally, an innocuous stimulus does not trigger a pain response in the brain. However, neuropathic pain, the sensory cortex of the brain incorrectly processes the stimulus as pain, which may show in a variety of ways. By increasing the applied mechanical stimulus and comparing to different control conditions, we may be able to see the effects of drug therapy with the BBB disruption. Though our rat experiments are still ongoing, I have been given the opportunity to continue my work at the lab for the rest of the summer with a global scholarship grant through the Focused Ultrasound Foundation. After we have collected data for more rats, I plan on analyzing the correlation between different brain regions through fMRI processing, as mentioned before. Localized drug delivery to specific targets in the brain is already used clinically by the FUS lab here at Brigham for chemotherapy delivery in glioblastoma patients. With this technology, doctors can increase the efficacy of an anti-cancer drug during chemotherapy. Focused ultrasound guided drug delivery could have a major impact on the field of cancer research, and as focused ultrasound technology advances, it may become a useful tool in disease treatment around the world. I would like to thank my mentor, Nick Todd, and our PI, Nathan McDonald, for this unique and exciting experience here at the Foos Lab.